In the summer of 2022, humanity activated the most powerful eye ever built to peer into the depths of space. The James Webb Space Telescope, a machine carrying mirrors coated in gold so thin you could see through them if they weren't backed by beryllium, opened its instruments and began to collect light from the universe. It was designed to see further than any telescope before it, to detect the faint infrared glow of the first galaxies, to peer through cosmic dust, to analyze the atmospheres of distant worlds. But no one, not the engineers who spent decades building it, not the scientists who proposed the observations, not even the most optimistic astrobiologists, expected what the telescope would find barely 2 years into its mission. Among the thousands of candidate worlds in the data queue, among the rocky planets and gas giants and ice worlds, there appeared a signal so compelling, so rich with the markers of biological activity, that the initial analysis team sat in stunned silence for nearly an hour before anyone spoke. The planet, orbiting a star we had never paid much attention to, exhibited atmospheric signatures that conventional models could not explain through geology alone, through volcanism alone, through any known abiotic process. The probability calculations, run and rerun, checked and triple-checked, kept returning the same range, 99.7%. 99.7% likelihood that what we were seeing required life to produce it. If you love deep space deep dives, you can subscribe. It helps more than you think. To understand the magnitude of what this means, we need to first understand what we mean by a probability of life, and why such a number is both astonishing and deeply troubling to calculate. Science does not deal in certainties when it comes to unverified discoveries. It deals in likelihoods, in confidence intervals, in the weight of evidence compared to alternative explanations. When scientists say there is a 99.7% chance that a planet hosts life, they are not saying they are certain. They are saying that, given the data they have, given every known non-biological explanation they can think of, given the chemistry and physics and geology they understand, the simplest explanation that fits all the observations is that something alive is producing the signals they see. But simplicity is not proof, and 99.7% is not 100%. That remaining 0.3% is vast, a chasm of uncertainty into which entire theories can disappear. The planet in question is designated K218b, though it has already acquired a dozen unofficial names in the press and among researchers. It orbits a red dwarf star called K218, located roughly 124 light-years from Earth in the constellation Leo. This is not far cosmically speaking. Light from this planet, light that left its atmosphere when the first humans were inventing the wheel, when agriculture was spreading across ancient Mesopotamia. That light is only now reaching us. We are seeing this world as it was in the deep past, but because stellar systems are stable on timescales of billions of years, there is every reason to think the planet still exists, still orbits, still holds whatever it is that produces the signals we detect. K280b is not like Earth. It is larger, with a radius about 2.6 times that of our planet and a mass roughly 9 times greater. This places it in a category astronomers call a sub-Neptune, a type of world that does not exist in our solar system. It is too large to be a rocky planet like Earth or Mars, but too small to be a gas giant like Neptune or Jupiter. The nature of sub-Neptunes is one of the great uncertainties in planetary science. Some may be rocky worlds with thick hydrogen atmospheres. Others may be ocean worlds, with global seas hundreds of kilometers deep overlying a rocky or icy core. Still others may be something in between or something we have not yet imagined. K2AB, based on its mass and radius, could fit any of these descriptions. But the data from James Webb suggests something more specific, something that has never been confirmed on any world beyond Earth. The telescope observed K218b using a technique called transmission spectroscopy. When the planet passes in front of its star, as seen from Earth, some of the starlight filters through the planet's atmosphere. Different molecules in that atmosphere absorb light at specific wavelengths, creating a kind of barcode, a unique signature that tells us what the atmosphere is made of. James Webb observed K218b during 5 separate transits, collecting data across a range of infrared wavelengths, and the results were striking. The atmosphere contains water vapor. This was expected because water is common in the universe. The atmosphere also contains methane. This was interesting, because methane is reactive and breaks down quickly under ultraviolet light from the star, so its presence suggests it is being replenished somehow. And then there was the third detection, the one that changed everything. Dimethyl sulfide, DMS, a molecule that, on Earth, is produced almost exclusively by life. Dimethyl sulfide is a simple organic compound, two methyl groups bonded to a sulfur atom. It smells like rotting cabbage, and it is released into Earth's atmosphere by phytoplankton, tiny marine organisms that float in the ocean and perform photosynthesis. When these organisms die or are eaten, they release DMS, which rises into the atmosphere and plays a role in cloud formation. On Earth, DMS is so closely associated with biological activity that its detection in the atmosphere is considered a strong biosignature, a sign that life is present. There are very few non-biological processes that produce DMS and none that produce it in the quantities we see on Earth. To find it in the atmosphere of an exoplanet, a world more than 100 light years away, is extraordinary. But the detection of a single molecule, even one as suggestive as DMS, is not enough to claim the discovery of life. Science requires multiple lines of evidence, redundancy, the ruling out of alternative explanations, and so the researchers did what they always do. They looked for ways they could be wrong. Could DMS be produced by volcanic activity? Possibly, but it would require very specific conditions, and there was no evidence of the other gases you would expect from volcanism, like sulfur dioxide in the quantities that should accompany it. Could it be produced by photochemistry, reactions driven by starlight in the upper atmosphere? The models said no, not in these amounts, not with this particular star. Could it be contamination, an error in the data, a ghost signal from the telescope itself? The data was checked, the calibrations reviewed, the observations repeated with different instruments. The signal remained. Could it be something else entirely? Some exotic chemistry we do not understand, some process unique to sub-Neptunes that has no parallel in our solar system? This is the hardest question to answer, because it asks us to imagine the unknown, to consider possibilities we have not yet thought of. And yet, even accounting for this, even granting that there might be processes we do not know about, the presence of DMS, combined with methane and water vapor in specific ratios, points most readily toward biological activity. The statistical models took all of this into account. They considered every alternative explanation, assigned probabilities to each, and calculated the likelihood that the observed data could be produced by non-biological processes. The result was 0.3%. 99.7% of the probability space was occupied by scenarios involving life. This does not mean scientists are 99.7% certain that life exists on K2-18b. It means that, based on current knowledge, the hypothesis that life is present is far more consistent with the data than any other single hypothesis. But current knowledge is always incomplete, and the history of science is filled with examples of unlikely explanations turning out to be correct. So what kind of life could exist on a world like K2-18b? The answer is deeply uncertain, but we can make educated guesses based on the conditions we infer. The planet orbits its star at a distance where liquid water could exist on the surface, in what astronomers call the habitable zone. But the habitable zone is a rough concept, defined by the distance at which a planet receives enough starlight to keep water liquid, assuming an Earth-like atmosphere. K2-18b's atmosphere is not Earth-like. It is thick, probably dominated by hydrogen and helium, with water vapor and methane mixed in. The surface pressure could be immense, dozens or even hundreds of times the pressure at Earth's sea level. Under such conditions, the boundary between atmosphere and ocean may not be sharp. Instead, there could be a gradual transition, a region where the atmosphere becomes so dense that it behaves like a fluid, and the ocean, if it exists, might be a supercritical fluid, neither fully liquid nor fully gas. If there is life in such an environment, it would not resemble life on Earth in any familiar way. It would not be forests and animals and birds. It would not be anything you could see with the naked eye, even if you could somehow stand on the surface without being crushed by the pressure or poisoned by the atmosphere. It would more likely be microbial, simple organisms floating in the ocean or drifting in the atmosphere, feeding on chemical energy or on light that filters down through the thick haze. These organisms, if they exist, would have evolved under conditions radically different from those on Earth. They would have adapted to high pressure, to low light, to an atmosphere rich in hydrogen. They might use biochemistry based on different molecules, different solvents, different energy sources. Or they might be surprisingly similar to Earth life, using water as a solvent, carbon as a structural element, drawing energy from redox reactions or from photosynthesis. We do not know. We cannot know without going there, without sampling the atmosphere or the ocean, without detecting the organisms directly. But the presence of DMS suggests that, whatever these organisms are, they are engaging in chemistry similar to Earth's phytoplankton. They are producing sulfur compounds as a byproduct of their metabolism, releasing them into the atmosphere, altering the chemical composition of their world. This is significant because it suggests a kind of life that is not just surviving, but thriving, present in sufficient numbers to affect the planet on a global scale. A few scattered microbes would not produce enough DMS to be detectable from 124 light-years away. To see this signal, there must be vast populations, ecosystems, possibly an entire biosphere. This is not a planet with life clinging to a few isolated niches. This is a planet where life, if the interpretation is correct, is a dominant force. The star K2-18 is a red dwarf smaller and cooler than the Sun. Red dwarfs are the most common type of star in the galaxy, making up about 70% of all stars. They burn their hydrogen fuel slowly, shining for trillions of years, far longer than stars like the Sun. This makes them attractive targets in the search for life, because their planets have more time for life to emerge and evolve. But red dwarfs also have drawbacks. They are prone to violent flares, sudden bursts of radiation and charged particles that can strip away a planet's atmosphere or sterilize its surface. They emit most of their light in the infrared, not the visible spectrum, which means that photosynthesis, if it occurs, would have to be adapted to lower energy photons, and their habitable zones are close in because the star is dim. A planet in the habitable zone of a red dwarf orbits much closer than Earth orbits the Sun, which means it is more likely to be tidally locked, with one side always facing the star and the other in permanent darkness. K2-18b may or may not be tidally locked. Its orbital period is about 33 days, which is short enough that tidal forces could have slowed its rotation until it became synchronous. If it is tidally locked, the climate would be extreme, with a permanent day side and a permanent night side. The day side would be hot, constantly bathed in starlight, while the night side would be cold, freezing whatever atmosphere could not circulate to the warmer hemisphere. Winds would howl between the two sides, driven by the temperature difference, creating storms of unimaginable violence. But if the atmosphere is thick enough, it could redistribute heat, warming the night side and cooling the day side, creating a more temperate environment across the planet. This is one of the many uncertainties. We do not know the planet's rotation rate. We do not know the thickness of the atmosphere in detail. We do not know the surface temperature or the wind patterns. We are working with indirect data, with models that make assumptions, and every assumption is a potential source of error. The detection of methane adds another layer to the puzzle. Methane is a greenhouse gas, much more effective at trapping heat than carbon dioxide. Its presence in the atmosphere would warm the planet, possibly enough to keep water liquid even at the relatively dim light levels provided by a red dwarf. But methane is also reactive. It breaks down in the presence of ultraviolet light, forming carbon dioxide and water. For methane to persist in the atmosphere, it must be replenished. On Earth, methane is produced by geological processes like volcanic outgassing and by biological processes like the digestion of organic matter by microbes. The methane in K2-18b's atmosphere could come from either source or from both, but the ratio of methane to carbon dioxide and the presence of DMS suggests that biology is a significant contributor. There is also the question of oxygen. On Earth, oxygen is produced by photosynthesis and is considered one of the strongest biosignatures because there are very few non-biological processes that produce free oxygen in large quantities. But James Webb did not detect oxygen in K2-18b's atmosphere, or at least not in amounts large enough to stand out above the noise. This is puzzling, because if there is photosynthetic life producing DMS, you might expect it to also produce oxygen. The absence of oxygen does not rule out life, but it complicates the picture. It suggests that, if there is life, it may not be photosynthetic in the way Earth life is, or that the oxygen is being consumed as fast as it is produced, reacting with methane or hydrogen or other gases in the atmosphere. Alternatively, the life on K2-18b might use a different energy source entirely. Not photosynthesis but chemosynthesis, drawing energy from chemical reactions rather than from light. The researchers have been cautious in their public statements, as they should be. Extraordinary claims require extraordinary evidence, and the claim that we have found life on another planet is about as extraordinary as it gets. The initial papers, published in peer-reviewed journals, lay out the data and the analysis, but they stop short of declaring the discovery of life. Instead, they present DMS as a candidate biosignature, a molecule whose presence is consistent with biological activity but not conclusive. They call for further observations, for more data, for the involvement of other telescopes and other techniques. They emphasize the uncertainties, the alternative explanations, the possibility that they are wrong. This is how science should work: caution rigor, the slow accumulation of evidence, the testing and retesting of hypotheses. But behind the caution, there is excitement. In private conversations, in emails, in conference hallways, scientists are allowing themselves to consider the possibility that this is real, that we have found what we have been looking for since humans first looked up at the stars and wondered if we are alone. The discovery, if confirmed, would be the most important in the history of science. It would answer a question that has been asked for millennia. It would tell us that life is not unique to Earth, that the processes that led to the origin of life here are not a singular fluke, but a natural outcome of chemistry and physics under the right conditions. It would mean that the universe is not a sterile void, but a place where life can and does arise, perhaps commonly, perhaps on billions of worlds across the galaxy, and it would raise 1,000 new questions. How did life arise on K2-18b? Is it related to life on Earth, perhaps seeded by panspermia, the transfer of life between worlds via meteorites or comets? Or did it arise independently, a second genesis, proof that life is a universal phenomenon? What is its biochemistry? Does it use DNA, RNA, proteins, the same molecular machinery that Earth life uses? Or does it use different molecules, different structures, different coding systems? Is it simple or complex? Is it confined to the ocean? Or has it colonized the atmosphere, the land if there is land, the subsurface if there is a solid surface at all? Is it intelligent? Almost certainly not, given the environment, but we cannot rule it out entirely. Intelligence is rare on Earth, confined to a few lineages that evolved under very specific conditions, but the universe is large and unlikely things happen given enough time and enough trials. The detection has also sparked debate about what to do next. Some argue that we should focus every available resource on K2-18b, that we should observe it at every possible wavelength, with every telescope we have, that we should design missions to study it in more detail. Others argue that we should be cautious, that we should not put all our eggs in one basket, that K2-18b might turn out to be a false alarm, and we should continue surveying other planets, building up a larger sample. There are plans to observe K2-18b again with James Webb, to collect more spectra, to search for other molecules that could confirm or refute the presence of life. There are proposals to use other telescopes, like the Hubble Space Telescope, which operates at different wavelengths, or ground-based observatories with adaptive optics that can partially compensate for atmospheric distortion. There are even early-stage discussions about future missions, space telescopes designed specifically to characterize the atmospheres of potentially habitable worlds, missions that would not launch for decades but could provide definitive answers. One of the challenges is that K2-18b is not an ideal target for detailed study. It is far enough away that even with James Webb, the signal is faint, the data noisy. The planet does not transit often, only once every 33 days, so each observation opportunity is precious. And the star itself, being a red dwarf, is relatively dim, which means there is less light to work with. To improve the signal, to detect weaker spectral features, to search for additional molecules, will require many more transits, many more hours of observation time, and observation time on James Webb is precious. The telescope is in high demand, with hundreds of research teams requesting time to study everything from distant galaxies to nearby asteroids. The team studying K2-18b has been allocated additional time, but it is not unlimited, and they must compete with other priorities. There is also the question of what other molecules to look for. DMS is a strong biosignature, but it is not the only one. On Earth, life produces a wide range of gases, some of which are present in the atmosphere in trace amounts but are detectable with sensitive instruments. Phosphine, for example, is produced by anaerobic bacteria and was controversially detected in the atmosphere of Venus in 2020, though that detection has since been disputed. Ammonia is produced by nitrogen-fixing bacteria. Nitrous oxide is produced by soil bacteria. If life on K280b is biochemically similar to Earth life, it might produce these gases as well. Alternatively, it might produce gases we have never heard of, molecules that are unique to its biochemistry, and we would have no idea what to look for. This is one of the frustrations of searching for life on other worlds. We are looking for what we know, for the signatures of Earth, like life, because that is all we have to go on. If life elsewhere is radically different, if it uses different chemistry, different molecules, we might not recognize it even if we see it. The discovery, if it holds up, also has philosophical implications. For centuries, humans have debated whether we are alone in the universe, whether Earth is special, whether life is a common or rare phenomenon. The Copernican principle, the idea that we are not at the center of the universe, that there is nothing special about our location or our time, suggests that life should be common. If the laws of physics and chemistry are the same everywhere, and if the building blocks of life are present throughout the universe, as they appear to be, then life should arise wherever conditions are suitable. But we have not found it, not until now, and the lack of evidence has led some to argue that life is rare, that Earth is a fluke, that we might be alone or nearly alone in the galaxy. The detection of life on K218b, if confirmed, would settle this debate, at least partially. It would show that life can arise on worlds very different from Earth, on sub-Neptunes with thick hydrogen atmospheres, on planets orbiting red dwarfs, under conditions that would be hostile to most Earth life. It would suggest that the origin of life is not a freak accident requiring very specific conditions, but a robust process that can occur in a variety of environments. And if life can arise on K218b, it can arise on countless other worlds. The galaxy contains hundreds of billions of stars, and most of those stars have planets. If even a small fraction of those planets are habitable, and if even a small fraction of habitable planets develop life, then the universe is teeming with biology, and we have been living in ignorance, alone not because life is rare, but because we have not yet looked hard enough. But the discovery also raises the Fermi paradox in a new light. If life is common, if it can arise on worlds as different as Earth and K218b, then where is everyone? Where are the intelligent civilizations, the radio signals, the megastructures, the signs of technology? The answer may be that life is common, but complex life is rare, or that intelligence is rare, or that technological civilizations are short-lived, destroying themselves or losing interest in expansion and communication. Or the answer may be that they are out there, but the distances are too vast, the timescales too long, the galaxy too large for us to have encountered them yet. Or the answer may be something we have not thought of, some factor we are missing, some aspect of the universe we do not yet understand. K218b is not the first exoplanet to show potential signs of life. Over the past few decades, astronomers have identified thousands of exoplanets, and many of them are in the habitable zones of their stars. Some have been studied with transmission spectroscopy, and a few have shown intriguing features. Water vapor has been detected on several exoplanets. Methane has been found on a handful, but none have shown the combination of molecules, the specific ratios and abundances that K218b displays. None have had a 99.7% probability assigned to the hypothesis of life. K218b is different, not because it is the only habitable world, but because it is the first where the evidence is strong enough to warrant serious consideration of the possibility that we are seeing biology in action. The planet was first discovered in 2015 by NASA's Kepler Space Telescope during its extended K2 mission. Kepler found the planet by detecting the slight dimming of the star's light as the planet passed in front of it, a technique called the transit method. The discovery was announced in 2016, and K218b was noted as being in the habitable zone, but little else was known about it at the time. Follow-up observations with ground-based telescopes measured the planet's mass using the radial velocity method, which detects the wobble of the star caused by the planet's gravity. The combination of mass and radius allowed astronomers to estimate the planet's density, which suggested it was not a purely rocky world, but had a significant envelope of gas. In 2019, the Hubble Space Telescope observed K2-18b and detected water vapor in its atmosphere, the first such detection on a planet in the habitable zone of its star. This was a significant result, and it made K2-18b a high-priority target for further study. But Hubble's instruments, powerful as they are, do not have the sensitivity to detect molecules like DMS that required James Webb with its larger mirror and its advanced infrared spectrographs. The observations of K2-18b by James Webb were conducted in 2023, and the data was released in late 2023 and early 2024. The detection of DMS was announced in a series of papers published in mid-2024, and the scientific community is still processing the implications. The announcement was carefully managed because the researchers were aware of the potential for misinterpretation and sensationalism. The phrase 99.7% chance of life is dramatic, and it is easy for the public to hear that and assume that life has been definitively found. But that is not what the number means. It is a probability, a statement about the relative likelihoods of different hypotheses given the data, and it is contingent on the assumptions built into the models. If one of those assumptions is wrong, if there is an exotic chemistry we have not accounted for, the probability could change. The researchers have been at pains to emphasize this, to explain that they are presenting evidence, not proof, and that more work is needed. But even with the caveats, the discovery is remarkable. It represents the culmination of decades of work, of building better telescopes, of developing better models, of searching for planets around other stars. The first exoplanet was discovered in 1992, orbiting a pulsar, a rapidly rotating neutron star. The first exoplanet around a sun-like star was discovered in 1995. At the time, the idea that we could detect exoplanets at all was considered optimistic, and the idea that we could study their atmospheres was science fiction. Today, we have found over 5,000 exoplanets, and we have analyzed the atmospheres of dozens of them. We have found hot Jupiters, gas giants orbiting close to their stars, so hot that their atmospheres are vaporizing. We have found super-Earths and mini-Neptunes, planets in size ranges that do not exist in our solar system. We have found planets orbiting binary stars, planets with orbits tilted at extreme angles, planets that are being torn apart by their star's gravity. The diversity of planetary systems is staggering, and it suggests that planet formation is a common and robust process, producing a wide range of outcomes depending on the initial conditions. K2-18b is one of the few exoplanets that is both in the habitable zone and has an atmosphere we can study in detail. Most exoplanets are too far from Earth, or their stars are too bright or too dim, or the planets themselves are too small or too large, or the geometry is wrong and the planet does not transit as seen from Earth. The planets we can study are a biased sample, and we must be careful not to overinterpret what we find. K2-18b may not be typical of habitable worlds. It may be an outlier, a special case, and the life it hosts, if it hosts life, may not be representative of life elsewhere. Or it may be typical, and the galaxy may be full of sub-Neptunes with thick atmospheres and microbial biospheres, and we just have not looked at enough of them yet to know. The atmosphere of K2-18b is also interesting from a climate perspective. The thick hydrogen envelope acts as a greenhouse, trapping heat and raising the surface temperature. But hydrogen is a light gas, and over time it can escape to space, especially for a planet orbiting close to a star. The planet's gravity is strong enough to retain hydrogen on timescales of billions of years, but not indefinitely. Eventually, the hydrogen will be lost, and the planet's atmosphere will change. What remains will depend on what lies beneath. If there is a deep water ocean, the water vapor could become the dominant atmospheric component, creating a steam atmosphere. If there is a rocky surface with volcanic activity, carbon dioxide and nitrogen could be released, creating an atmosphere more like Venus or Earth. The long-term evolution of K2-18b is uncertain, and it depends on factors we do not fully understand, like the rate of atmospheric escape and the rate of volcanic outgassing. There is also the question of how the planet formed. Sub-Neptunes are thought to form either as rocky cores that accumulate hydrogen from the protoplanetary disk, or as ice-rich bodies that migrate inward from the outer regions of the planetary system. K2-18b's composition suggests it has a significant amount of water, possibly in the form of a global ocean overlying a rocky core. This water could have been delivered by comets and asteroids during the planet's formation, or it could have been present in the material from which the planet formed. The exact formation history is uncertain, and it is one of the many things we would like to understand better. The detection of life on K2-18b, if it is confirmed, will also have implications for how we search for life elsewhere. It will tell us that we should focus on sub-Neptunes, not just Earth-sized rocky planets. It will tell us that red dwarf stars, despite their flares and their dim light, can host habitable planets. It will tell us that thick hydrogen atmospheres, far from being hostile to life, may actually help by providing a greenhouse effect that keeps the planet warm. And it will tell us what molecules to look for, what combinations of gases are most indicative of biology. This will inform the design of future missions, the selection of targets, the development of instruments. But there is also the possibility that the detection is wrong, that the DMS signal is not what it appears to be, that there is some non-biological explanation we have not considered. This is why independent verification is so important. Other teams, using other telescopes, will observe K2-18b and check the results. If they confirm the detection of DMS, if they find the same ratios of gases, if they rule out the alternative explanations, then the probability will increase. If they do not, if the signal disappears or changes, then the interpretation will need to be revised. This is how science works. It is a process of iterative refinement, of testing and retesting, of building consensus through independent verification. The public reaction to the announcement has been mixed. Some people are excited, hopeful, thrilled at the possibility that we are not alone. Others are skeptical, pointing out that we have been fooled before, that there have been other claims of biosignatures that turned out to be false alarms. Still others are indifferent, focused on more immediate concerns, on problems here on Earth. And there are those who are fearful, worried about what the discovery of alien life might mean, whether it poses a threat, whether it changes our place in the universe in ways we are not prepared for. These reactions are all understandable. The discovery of extraterrestrial life, even microbial life on a distant planet, would be a profound event, and it is natural for people to have strong feelings about it. From a scientific perspective, the discovery, if confirmed, would be the beginning, not the end. It would open up an entire new field of study, the biology of K2-18b, and by extension, the biology of other worlds. We would want to know everything we could about this life. How does it reproduce? How does it metabolize? What is its genetic material, if it has any? How does it interact with its environment? Is it a single species, or are there many species and ecosystem? Is it simple or complex? Has it evolved over billions of years, or did it arise recently? These questions cannot be answered from Earth, not with current technology. They require missions probes, perhaps even sample return, though the distance makes that extraordinarily difficult. K2-18b is 124 light-years away. With current propulsion technology, a spacecraft would take tens of thousands of years to get there. Even if we could accelerate a probe to a significant fraction of the speed of light, which we cannot, it would still take over a century to reach the planet, and the data would take another century to return. This is not an insurmountable problem, but it is a severe constraint. We will not be visiting K2-18b in person, not in our lifetimes, not in our grandchildren's lifetimes, not for many generations. What we can do is continue to observe it from a distance, to refine our measurements, to develop better models, to learn as much as we can from the light that reaches us. There is also the ethical question of what we should do if we confirm that K2-18b hosts life. Do we have a responsibility to protect it, to avoid contaminating it with Earth life if we ever do send a probe? Do we have a right to study it, to interfere with it, to potentially disrupt it? These are questions humanity has grappled with in the context of Mars and other bodies in our solar system, where we have planetary protection protocols to minimize the risk of contamination. But K2-18b is different. It is so far away that contamination by accident seems impossible. If we ever sent a mission there, it would be deliberate, carefully planned. and we would have time to think through the ethical implications. But those implications are not trivial. If there is life on K2-18b, even simple microbial life, it has value, intrinsic value, independent of its utility to us. And we have an obligation to consider that value when we make decisions about how to interact with it. The discovery also raises theological and philosophical questions for many people. What does it mean for humanity if we are not unique, if life exists elsewhere? Different religious traditions have different answers. Some see no conflict, arguing that a creator capable of making one world with life is capable of making many. Others see it as a challenge, a test of faith, a new revelation. Still others see it as irrelevant, a scientific fact that does not touch on matters of ultimate meaning and purpose. These are deeply personal questions, and there is no single answer that will satisfy everyone. But they are worth thinking about, because the discovery of life on another world would change our understanding of ourselves, of our place in the cosmos, of what it means to be human. For scientists, the discovery would be a validation of the search for extraterrestrial life, a field that has sometimes been dismissed as speculative or unlikely to produce results. For decades, researchers have looked for signs of life on Mars, on the moons of Jupiter and Saturn, in the atmospheres of exoplanets, and they have found tantalizing hints but no definitive proof. K2-80b, if the interpretation holds, would be the proof, the confirmation that the search is not in vain, that life is out there waiting to be found. It would justify the resources invested in telescopes and missions, the careers spent analyzing data, the years of patient observation, and it would inspire a new generation of scientists, young people who will grow up knowing that life exists beyond Earth, who will take for granted what their grandparents could only imagine. The technical challenges of studying K2-18b are formidable. The planet's atmosphere is complex, with multiple layers, multiple chemical species, interactions between gases that are not fully understood. The models used to interpret the spectra are sophisticated, but they rely on assumptions about temperature structure, pressure profiles, cloud cover, and atmospheric dynamics. Any of these assumptions could be wrong, and if they are, the interpretation of the data could change. The researchers are working to refine the models, to test different scenarios, to see which ones best match the observations. This is painstaking work requiring expertise in chemistry, physics, astronomy, and computer modeling. It is the kind of work that does not make headlines, but it is essential for understanding what we are seeing. One of the uncertainties is the role of clouds. Clouds can obscure spectral features, making it harder to detect molecules in the atmosphere. They can also create false signals if the cloud composition mimics the spectral signature of a particular molecule. K2-18b likely has clouds because water vapor is present and the temperature is right for water droplets or ice crystals to condense. But we do not know how thick the clouds are, how high they extend, or what they are made of. This uncertainty affects the interpretation of the spectra, and it is one of the reasons why the researchers are cautious about their conclusions. Another uncertainty is the planet's interior structure. Is there a solid surface beneath the atmosphere, or does the atmosphere transition gradually into a liquid or supercritical ocean? If there is an ocean, how deep is it? Does it reach down to a rocky core, or is there a layer of high-pressure ice between the ocean and the core? These questions matter because they affect the planet's heat budget, its magnetic field if it has one, and the potential for volcanic or tectonic activity. A planet with active geology could release gases into the atmosphere that mimic biological signatures, and we need to understand the geology to interpret the chemistry. The planet's rotation rate is also unknown. If K2-18b is tidally locked, the climate would be very different from a planet that rotates. Tidal locking would create extreme temperature gradients, but it would also create strong winds, and those winds could redistribute heat and gases, creating a more uniform atmosphere. If the planet is not tidally locked, the day-night cycle would drive different dynamics, possibly including weather patterns similar to those on Earth, with storms and seasons and circulation cells. We do not know which scenario is correct, and without more data, we cannot determine it. There is also the question of magnetic fields. Earth has a strong magnetic field, generated by the motion of molten iron in its core, and this field protects the atmosphere from being stripped away by the solar wind. K2-18b may or may not have a magnetic field, depending on its interior structure and rotation rate. If it does not, the atmosphere could be gradually lost to space, especially given the planet's proximity to its star. But the thick hydrogen envelope might provide enough mass that the loss is negligible on timescales of billions of years. Or the planet might be replenishing its atmosphere through volcanic outgassing or through the release of gases from the ocean. These are all possibilities, and we do not have enough data to distinguish between them. The researchers are planning follow-up observations to address some of these uncertainties. They want to observe the planet at different phases, not just during transits, to see if the atmospheric composition changes as different parts of the planet rotate into view. They want to observe at different wavelengths to detect additional molecules and to constrain the temperature and pressure profiles. They want to observe over longer timescales to see if the signals vary with time, which could indicate seasonal changes or volcanic eruptions or other dynamic processes. All of this will take time, and it will require careful planning and coordination, because telescope time is limited and there are many other worthy targets competing for attention. In the meantime, theoretical work is ongoing. Modelers are running simulations of K2-18b's atmosphere, trying to reproduce the observed spectra and to predict what else we might find. They are exploring different scenarios, different assumptions, different chemical networks, to see which ones are most consistent with the data. They are also thinking about what kinds of life could exist under the conditions inferred for K2-18b, what metabolisms would be possible, what biochemistry might work. This is speculative, but it is informed speculation, grounded in our understanding of chemistry and biology and thermodynamics. One intriguing possibility is that life on K2-18b could be fundamentally different from Earth life, using different molecular machinery, different solvents, different energy sources. Earth life is based on carbon, uses water as a solvent, and relies on DNA and RNA for information storage and proteins for catalysis. But there is no reason why life elsewhere must use the same molecules. Carbon is abundant and versatile, but silicon is also abundant and can form complex molecules. Water is a good solvent, but ammonia or methane could work under different conditions. DNA is an elegant solution to the problem of information storage, but other molecules could serve the same function. If life on K2-18b is radically different, it would be a second example of life, a second data point in our understanding of what life is and how it works. And that would be as valuable, scientifically, as the discovery itself. But if life on K2-18b is similar to Earth life, that would also be remarkable, because it would suggest that there are constraints, that chemistry and physics channel the evolution of life in particular directions, that the solutions to the problems of survival and reproduction are limited. It would suggest that carbon-based, water-using life is not an accident of Earth's history, but a natural outcome of the laws of nature. This would have implications for how common life is in the universe, because if the same solutions arise independently on different worlds, then life may be more predictable, more inevitable than we thought. The discovery has also prompted renewed interest in the concept of biosignatures, the signs that life leaves in the atmosphere or on the surface of a planet. Oxygen is the classic biosignature, because on Earth it is produced by photosynthesis and would quickly disappear if life stopped producing it. But oxygen is not the only biosignature, and it may not be the best one. Methane in the presence of oxygen is a strong biosignature because the two gases react with each other and would not coexist in large quantities without a source that replenishes them. DMS is another, as is phosphine. There are also surface biosignatures, like the red edge, a sharp increase in reflectance at near-infrared wavelengths caused by chlorophyll in plants. None of these signatures is foolproof, and all of them can be mimicked by non-biological processes under certain conditions. But in combination, they can build a strong case for the presence of life. The challenge is that we are limited by what we can observe. We cannot see the surface of K2-18b directly, because it is too far away and too faint. We can only study the atmosphere, and only the upper atmosphere at that, the region where starlight passes through during a transit. The lower atmosphere and the surface, if there is a surface, are hidden from view. This limits what we can learn, and it means we must be cautious about our conclusions. We are interpreting a few spectral lines, a handful of data points, and from those we are trying to infer the presence of life the nature of the environment, the history of the planet. This is ambitious and it is fraught with uncertainty, but it is the best we can do with current technology. Future telescopes will help. The Habitable Worlds Observatory, a proposed NASA mission, would be specifically designed to image and characterize Earth like planets around nearby stars. It would have a larger mirror than James Webb, and it would operate at optical and ultraviolet wavelengths, where many biosignatures are more easily detected. It would be able to take direct images of planets, separating their light from the glare of their stars, and it would be able to study their atmospheres in much greater detail. But the Habitable Worlds Observatory is still in the planning stages, and it will not launch for at least 2 decades, possibly longer. In the meantime, we are reliant on James Webb, Hubble, and ground-based telescopes, all of which have limitations. There is also the European Extremely Large Telescope, currently under construction in Chile, which will have a mirror 39 meters across, the largest optical telescope ever built. It will be able to study exoplanet atmospheres in unprecedented detail, though it will be most effective for planets around nearby stars brighter than K2-18. And there are other missions in development, like the Nancy Grace Roman Space Telescope, which will search for exoplanets using microlensing and direct imaging, and the Ariel mission, which will study the atmospheres of hundreds of exoplanets to understand their diversity. The field of exoplanet science is advancing rapidly, and each new mission brings new capabilities, new discoveries, new insights. The discovery of life on K2-18b, if confirmed, would also have implications for the Drake equation, a formula proposed by astronomer Frank Drake in the 1960s to estimate the number of communicative civilizations in the galaxy. The equation multiplies together a series of factors: the rate of star formation, the fraction of stars with planets, the number of habitable planets per star system, the fraction of habitable planets where life arises, the fraction of life-bearing planets where intelligent life evolves, the fraction of intelligent species that develop technology capable of communication, and the length of time such civilizations survive. Most of these factors are highly uncertain, and the equation is more a framework for thinking about the problem than a tool for making precise predictions. But the detection of life on K2-18b would provide a hard data point for one of the factors: the fraction of habitable planets where life arises. If life can arise on K2-18b, a world quite different from Earth, then that fraction may be higher than pessimists have assumed, and the number of life-bearing planets in the galaxy could be large. But the equation also highlights how little we know about the other factors, especially the fraction of life-bearing planets where intelligent life evolves, and the fraction of intelligent species that develop technology. On Earth, life arose relatively quickly, within a few 100 million years after the planet cooled, but intelligence took billions of years to evolve, and technological civilization has existed for only a few thousand years. If this is typical, then intelligence may be rare, even if life is common, or Earth may be unusual, and on other worlds intelligence may arise more readily, or more slowly, or not at all. We do not know, and without more examples, we cannot say. The question of whether K2-18b hosts intelligent life is almost certainly no. The environment is harsh by our standards, with high pressure, low light, and a thick atmosphere that would make the development of complex, multicellular life, let alone intelligence, extremely difficult. Intelligence on Earth evolved in an environment where vision was useful, where mobility was advantageous, where communication and tool use provided benefits. On K2-18b, floating in a dark ocean or drifting in a thick atmosphere, those advantages would be less clear. It is possible that some form of complex life could exist, but intelligence seems unlikely. And even if it did exist, the chances that it would develop technology capable of communication are vanishingly small. So while K2-18b may host life, it is almost certainly not a place where we will find a signal, a message, a counterpart to humanity. But the discovery would still be transformative, because it would show that the origin of life is not a miracle, not a one-time event, but a repeatable process. And if it can happen twice, it can happen countless times. The galaxy may be full of life, microbial life, simple life, life that goes about its business of metabolism and reproduction without ever developing the complexity, the awareness, the curiosity that characterizes humanity. And that would be a strange and humbling realization, To know that we share the universe with countless other forms of life, none of which know we exist, none of which care. The philosophical implications of this are profound. For most of human history, we have assumed we were special, chosen, unique. The discovery of extraterrestrial life would force us to reconsider that assumption, to accept that we are one example among many, that life is not a privilege but a common feature of the universe. This does not diminish our significance, but it changes it. We are not the center of creation, but we are part of something larger, something grander. We are participants in a cosmic story that extends far beyond our planet, our star, our galaxy. And that is, in its own way, more meaningful than being alone. The discovery would also raise practical questions. If life exists on K2-18b, should we try to communicate with it? Even though it is almost certainly non-intelligent. Should we send probes to study it? Even though the distance makes that extraordinarily difficult. Should we protect it? Designate it as off-limits, ensure that we do not contaminate it or disrupt it. These are questions we will need to answer, and they will require input from scientists, ethicists, policymakers, and the public. There is no handbook for how to handle the discovery of extraterrestrial life, no established protocols, no international consensus. We will be making it up as we go, guided by our values, our knowledge, and our best judgment. The history of the search for life beyond Earth is a story of persistent hope and repeated disappointment. For centuries, people believed that Mars was inhabited, that the canals seen on its surface were evidence of an ancient civilization struggling to survive on a dying world. The canals turned out to be optical illusions, artifacts of the human tendency to see patterns where none exist. In the mid-20th century, scientists thought that Venus might be a lush jungle world, warm and wet beneath its thick clouds. The reality, revealed by Soviet probes in the 1960s and 70s, was far more hostile: a surface hot enough to melt lead, an atmosphere of carbon dioxide thick enough to crush a submarine, clouds of sulfuric acid. The moons of Jupiter and Saturn offered new hope, with their subsurface oceans and potential for chemosynthetic life, but so far no definitive signs have been found. The search for life on exoplanets is a more recent development, made possible by the discovery of the first exoplanets in the 1990s. For the first decade or so, the exoplanets we found were not promising candidates for life. They were hot Jupiters, gas giants orbiting so close to their stars that their atmospheres were boiling away, or they were planets in highly eccentric orbits, swinging from freezing cold to scorching hot with each pass around their star. But as detection techniques improved, we began finding smaller planets, rocky worlds, some of them in the habitable zones of their stars. The Kepler mission, launched in 2009, revolutionized the field by discovering thousands of exoplanets, including many Earth-sized worlds. But Kepler could only detect planets, not characterize them in detail. That required follow-up observations with other telescopes, and those observations have been slow and painstaking. K2-18b was one of the Kepler discoveries, and it has taken nearly a decade of follow-up work to reach the point where we can seriously discuss the possibility of life. This is typical of how science progresses. It is not a series of dramatic breakthroughs, but a slow accumulation of data, a gradual refinement of models, a patient testing of hypotheses. The detection of DMS is a breakthrough, if it holds up, but it is built on years of prior work, on the development of new instruments, on the careful calibration of telescopes, on the theoretical understanding of atmospheric chemistry. The James Webb Space Telescope itself is a marvel of engineering, the product of decades of planning and construction. The telescope's primary mirror is 6.5 meters across, made up of 18 hexagonal segments, each coated with a thin layer of gold to maximize its reflectivity at infrared wavelengths. The mirror is so large that it had to be folded up to fit inside the rocket that launched it, and then carefully unfolded in space, a process that took months and involved hundreds of precise movements. The telescope orbits the Sun at a point called L2, about 1.5 million kilometers from Earth, where it is shielded from the Sun's heat by a multi-layered sunshield the size of a tennis court. This allows the instruments to stay cold, which is essential for detecting faint infrared signals. The telescope's spectrographs are among the most sensitive ever built, capable of detecting the faint absorption lines left by molecules in the atmospheres of distant planets. These instruments split the light into its component wavelengths, creating a spectrum that can be analyzed to determine the chemical composition of the source. The process is delicate and requires exquisite precision. The planet K280b is so far away that the amount of light we receive from it is infinitesimal, and the fraction of that light that passes through the planet's atmosphere during a transit is even smaller. To detect molecules like DMS, the instruments must be able to pick out tiny differences in the spectrum, differences that amount to a few parts per million or even less. This requires not only sensitive detectors, but also careful calibration, long exposure times, and sophisticated data analysis. The data analysis itself is a major undertaking. The raw data from the telescope is not immediately useful. It must be processed to remove instrumental artifacts, to correct for the motion of the telescope, to subtract the background noise, to account for variations in the star's brightness. Only then can the spectrum be extracted and analyzed, and even then, the interpretation is not straightforward. The spectrum contains the signatures of many molecules, and they overlap and blend together, making it difficult to pick out individual components. The researchers use complex models to fit the observed spectrum, adjusting the abundances of different molecules until they find the combination that best matches the data. This is an iterative process, and it can take months or even years to arrive at a definitive result. The detection of DMS is particularly challenging because it is present in small amounts, and its spectral signature overlaps with those of other molecules. The researchers had to carefully disentangle the different contributions, using what they know about the chemistry of the atmosphere to guide their interpretation. They also had to rule out the possibility that the signal is an artifact, a false detection caused by noise or by some quirk of the data processing. This required multiple independent analyses using different techniques and different assumptions to ensure that the result is robust. The fact that the signal has held up under this scrutiny is encouraging, but it is not definitive. There is always the possibility that something has been overlooked, that some assumption is wrong, that there is an alternative explanation that has not been considered. This is why the researchers are calling for more observations, more data, more independent confirmation. Only when the result has been replicated by other teams using other instruments can we be confident that it is real. But if it is real, if K218b does host life, then the implications extend far beyond this one planet. It would tell us that life is not confined to Earth like worlds, that it can arise in a wide range of environments, that the universe is more hospitable to biology than we might have thought. It would also tell us something about the nature of life itself, about what is essential and what is contingent. Earth life uses a particular set of molecules, a particular biochemistry, but is that the only way? Or are there other possibilities, other solutions to the problem of life that we have not yet imagined? These are not idle questions. They go to the heart of what life is, how it arises, how it evolves. On Earth, all life shares a common ancestor, a single organism that lived billions of years ago, and from which all modern life is descended. This means that all Earth life uses the same basic biochemistry, the same genetic code, the same molecular machinery. We cannot tell from studying Earth life alone which features are universal and which are accidents of history. But if we find life on K218b, and if it uses different biochemistry, then we will have a second data point, a way to compare and contrast, to see what is essential and what is optional. This is one of the reasons why the search for life beyond Earth is so important. It is not just about finding aliens, though that would be exciting. It is about understanding life itself, about learning what makes life possible, what constrains it, what shapes its evolution. Every new example of life we find will teach us something new, will expand our understanding, will challenge our assumptions. And K218b, if it hosts life, will be the first such example, the first time we have found life that did not originate on Earth. The discovery would also have practical implications. If we can understand how life arises on different worlds, we can make better predictions about where else to look. We can design more effective missions, target more promising candidates, develop better instruments. We can also learn about the limits of life, about how extreme the conditions can be and still support biology. This has implications for our search for life in our own solar system, on places like Mars or Europa or Enceladus, where the conditions are harsh but not necessarily impossible. Mars, for example, was once much wetter and warmer than it is today, and it may have hosted life in its ancient past. If it did, then there may be fossils preserved in the rocks, or perhaps even living microbes surviving in protected niches underground. Europa and Enceladus are icy moons with subsurface oceans, kept liquid by tidal heating. These oceans are in contact with rocky cores, which could provide the minerals and chemical energy needed for life. Both moons have geysers that erupt from their surfaces, shooting water and other materials into space, and these geysers could be sampled by a spacecraft to search for signs of life. The search for life in our solar system is complementary to the search for life on exoplanets. The advantage of searching nearby is that we can send probes, we can land on the surface, we can drill into the ice, we can collect samples and bring them back to Earth for detailed analysis. The disadvantage is that there are only a few targets, and if they do not host life, we are out of luck. The advantage of searching exoplanets is that there are thousands of them, and if one does not host life, we can move on to the next. The disadvantage is that they are far away, and we can only study them from a distance, relying on the faint light that reaches us. Ideally, we would do both. We would search for life in our solar system and on exoplanets, using every tool at our disposal, maximizing our chances of success. And that is what we are doing. Missions to Mars, Europa, and Enceladus are in various stages of planning and development, and telescopes like James Webb are studying exoplanets. The discovery of life on K2-18b, if confirmed, would not end the search. It would intensify it, providing motivation and direction for future efforts. There is also the question of how the discovery would affect our culture, our art, our literature, our sense of ourselves. The idea of alien life has been a staple of science fiction for over a century, inspiring countless stories, films, and television shows. But these are works of imagination, not reality. The discovery of real alien life, even if it is just microbes on a distant planet, would be different. It would make the universe feel more alive, more connected, less empty. It would change the way we think about our place in the cosmos, the way we tell stories about ourselves. Some have argued that the discovery of extraterrestrial life would be a unifying event for humanity, a reminder that we are all part of the same planet, the same species, facing the same challenges. Others have argued that it would be divisive, that different groups would interpret it in different ways, that it would become another source of conflict. The truth is probably somewhere in between. The discovery would mean different things to different people, and that is okay. What matters is that we engage with the discovery thoughtfully, that we consider its implications, that we use it as an opportunity to reflect on what it means to be human. In the end, the question of whether K2-18b hosts life may take years to answer definitively. The data is compelling, but it is not conclusive. More observations are needed, more analysis, more modeling, more debate. The scientific process is slow and deliberate, and that is as it should be. The stakes are too high for hasty conclusions. But if the answer is yes, if we do confirm that K2-18b hosts life, then we will look back on this moment as one of the great turning points in human history. The moment when we learned that we are not alone, that life is a universal phenomenon, that the cosmos is far more wondrous and strange than we ever imagined. And perhaps, in the end, the strangest thing about the universe is not that it contains life, but that it contains beings like us, capable of asking these questions, capable of building machines to peer across the void, capable of finding meaning in spectra and statistics, capable of wonder. We are the universe looking at itself, trying to understand itself, and in that act of looking, of questioning, of reaching toward the unknown, we fulfill a purpose that may be as rare and precious as life itself. The discovery of life on K2-18b would not diminish this purpose. It would enhance it by showing us that we are part of something larger, something that extends across the stars, something that connects us to the deepest mysteries of existence. And that connection, that sense of participation in a cosmic story, is worth pursuing, no matter how long it takes, no matter how difficult the journey.